Hi. Oops.
Hi, my name is Antonio Franze. I am postdoc in Susanna Mindenduur lab in URI. And I will present a poster on the temperature response of heterotrophic protists to temperature. We did experiment both with culture, established culture in our lab, and with the new isolates from Narragansett Bay. The main goal is to try to to obtain rates over a really wide range of temperature from 0 to 25, 22 degrees, and with the main objective to collect data, particularly at a lower temperature, which has been perpetrated the idea that heterotrophic protists are really affected by lower temperature for their growth and grazing. So we are going to demonstrate how those guys can actually, if they are adapted to colder temperature, they can actually grow pretty well and graze, and this is, has big effect on our modeling effort for the carbon export in the ocean.
Anyone else?
I'm another delinquent postdoc who didn't send slides. I'm Karen Stamieszkin. I'm a postdoc with Deb Steinberg down at VIMS. And also, most recently before that was a postdoc at Bigelow Lab. I study carbon export, mainly through zooplankton-related pathways. I do both field observations, lab experiments, and I really enjoy modeling, mostly trait-based mechanistic type modeling. I've also studied phytoplankton phenology and trying to connect changes in phenology and large-scale patterns in ocean phytoplankton phenology to temperature, zooplankton, and other higher trophic levels in terms of their seasonality and seasonal cycles. I'm sure I'm missing something. Oh, and my poster. Come see my poster. It's on fertilization of the upper ocean by different terrigenous sources of iron, including volcanic ash. Thank you.
Hey, I'm Matthias Oddo. I finished my undergrad in 2016. From then on, for 2 years, I was working in a company that's developing ocean fertilization stuff, specifically iron fertilization, offshore Canada and also Peru. I quit that in May, kind of bad terms. And I'm working in the grad lab of Maite Maldonado in UBC. I'll be doing research on the risks of iron fertilization. So it's a whole 180. And also ecosystem modeling with the ecopath model. And I just finished, on the way here, was my thesis for that course, an ecopath model for assessing the impact of sudden enrichment of phytoplankton and how that would impact specifically the Haida-Gwaii ecosystem. And it turned out quite interesting. So there was a project, if you know about it, the Haida project from 2012. They dumped iron. It was very controversial. They claimed 2 years later that salmon populations were recovered. The link is very hazy, though my model kind of shows that. So that was interesting. It has a lot of assumptions and it's very coarse. But yeah, it's what I'm doing, and I'm beginning. So see how it goes.
So I didn't prepare a slide either. I'm a postdoc. My name is Françoise Morison. I'm a postdoc in Mindenduur lab at URI Graduate School of Oceanography. And I've spent a lot of time during my PhD and also during this past year and a half as a postdoc trying to constrain grazing rates of microzooplankton in the open ocean. I've done a lot of work in the North Atlantic, recently in a NAMES project, where we've accumulated a lot of really nice data to try and constrain the losses due to grazing for the entire mix layer by incubating water from the surface, which is assumed to represent the mix layer at a bunch of different light levels. So that data now is available to enable modeling for the mix layer and relate those estimates to the ones that we can infer from satellite data, which are the basis of the new hypothesis of this interaction between grazers and phytoplankton that are driven by these changes in the mix layer and that have evolved into that disturbance recovery hypothesis, which ends up governing the start date of the spring phytoplankton bloom in the North Atlantic during the middle of the winter instead of what has been thought before during spring. I've also done a lot of measurements in Antarctica, and we've found some interesting responses. We measured microzooplankton grazing rate at 2 opposite seasons, once during late fall and another time during early summer along the western Antarctic Peninsula. And we actually found similar responses across the seasons and that were unrelated to the availability of prey that was there. So I have a poster today showing some of the results of the NAMES project, and I hope you will come and see me.
Hi, my name is Jia Liu. I'm a postdoc at Princeton and also visiting scientist at JFDL. I work with Sir Charlie Stock this year and John Dang. So this evening I'm going to present a poster on the role of river nutrient loading on the coastal ocean, everywhere, global coastal ocean. So if your area has rivers, and please come and see my poster. So we ran a fairly high resolution ocean model, which is core degree. We also have a higher resolution 8th degree model we're running. And we have a land model, process land model that link hydrological cycle and soil and vegetation growth that provide us time varying nutrient river loading, dissolved organic nitrogen. So that is offline linked to our ocean model. So we ran a suite of sensitivity experiment to test the coastal chlorophyll and hypoxia oxygen and the sensitivity of those responses to the variability in river nutrient loading. So please come and see my poster this evening. Thank you.
Like everyone is doing, so I have to do it. My name is Nicolas Mayot. I'm currently a postdoc at the Bigelow Laboratory. And before that, I've done my PhD in France, in Villefranche-sur-Mer, with Hervé Claustre and Fabricio D'Ortenzio, working on biochemical argo floats in the Mediterranean Sea. And I'm in the lab with Patty Matrai, and I'm working with biochemical argo floats and model in the Greenland Sea and the Norwegian Sea, so in this area. And I'm interested in understanding the phytoplankton seasonality in the seasonal high zone in this area. And tonight and during the week, I'm going to present a poster of data of biochemical argo floats able to sample under the ice. And so we have a nice, we saw some nice features about the ice edge bloom and also after during the summer. And so if you want to have more information about it, just stop by. Thank you.
All right. Hi, everyone. I'm Bianca Cruz. I'm from Arizona State University, a PhD student under Susanna Neuer's lab. We essentially studied the mechanisms by which picophytoplankton contribute to the biological carbon pump In the lab, we look at mechanisms such as the role of ballasting minerals, heterotrophic bacteria interactions, as well as the role of exopolysaccharides. Tonight I'll be presenting a poster on the molecular and microscopic characterization of sinking particles at the Bermuda Atlantic Time Series Station. And I have essentially divided different particle types into like phytodetrital aggregates or fecal pellet nucleated aggregates, which could potentially help us elucidate the mechanisms by which these phytoplankton may contribute to export.
Hi everyone, I'm not a postdoc anymore. The good news is that I just got a permanent position at the, thank you. Yes. At the University of São Paulo, Brazil. I did part of my PhD here at RUI, so I feel home and I feel very thankful for the opportunity. So I'm interested in the role of chemosynthesis in the carbon cycle. So I study chemosynthesis by combining C14 incorporation and combining with molecular tools to study microbial diversity and functionality. So I'm starting a new lab, I'm writing new projects, I will have students, I hope, and I'm looking for new collaborations. So I'm very open to talk to you all. Thanks for the opportunity.